Since the emergence of life over 2 billion years ago, our DNA has been our destiny. Our genetic information dictates how we look, how our bodies work, and even how we interact with the world. Our DNA shapes who we are, but we may be at the dawn of a new age, the era where we can change our DNA at will. We could cure millions suffering from genetic disorders, but also open the door to a dark chapter in human history. This is the story of one of the biggest discoveries of the 21st century. This is the story of CRISPR gene therapy. Our story begins in 1962, Wisconsin, United States. Scientist William Schabalski is growing cancer cells in a petri dish. These cancer cells though were special, as they lacked a gene called HRPT. This made it impossible for the cancer cells to grow in a specific medium. However, when Schabalski supplemented the cancer cells with HRPT DNA, they gained the ability to grow in the medium. Somehow the cancer cells integrated the supplemented DNA into their own genetic information. This was the first time in history someone had directly edited DNA in a cell. And it sparked a revolutionary idea. If we somehow figure out how we can deliver DNA into an organism, could we cure genetic diseases? You see, a genetic disease is a disease where our DNA has changed and mutated so that certain cells cannot perform certain functions anymore. If the CFTR gene, for example, is destroyed, we cannot properly import or export water and salts into certain cells anymore. This causes a sticky mucus to build up in the pancreas and the lungs, and we get cystic fibrosis. However, if we supplement the right cells with the CFTR gene, then they should become functional again and we could cure cystic fibrosis. And beyond cystic fibrosis, there are over 7,000 other genetic disorders, all of which could be cured when we deliver the right genes to the right cells. The race to develop the first gene therapy was on. 1970, Tennessee, United States. A team led by Dr. Stanford Rogers aimed to treat 2 girls who had a serious genetic disorder caused by a mutation in a gene responsible for metabolizing the amino acid arginine. To do this, they injected the show papillomavirus, which they had previously shown could alter arginine levels in cells. The trial was a failure, but it grabbed media attention, leading to more investments in gene therapy and the first true success. 1990, LA, United States. French Anderson, Michael Blaise, and Kenneth Calvert treated a young girl who suffered from ADA-SCID, a traumatic immune deficiency caused by a missing gene called adenosine deaminase. The doctors isolated bone marrow stem cells from the girl and transfused them with a virus that harbored the missing gene. Then they transplanted the cells back into the patient. Surprisingly, the girl suffered no severe side effects and was cured. This was the first successful gene therapy, and it launched the gold rush. Pharmaceutical companies and startups alike started to develop treatments against numerous genetic diseases. It felt like we were on the verge of curing any form of genetic disease. But then something happens. 1999, Pennsylvania, United States. 18-year-old Jesse Gelsinger volunteered for a safety trial. He suffered from a genetic disease that leads to the buildup of ammonia in the liver. The doctors injected an adenovirus into him that contained the intact gene the man needed. Within hours after the injection, Gelsinger developed severe side effects. His immune system went into a catastrophic overdrive, and 4 days later, he passed away from multi-organ failure. The scientists underestimated how his body would react to the treatment. The man's body recognized the virus as a foreign entity, and his immune system went ballistic. Then a second tragedy struck. In 2002, infants suffering from a severe immune disorder were treated with gene therapy. But years after, several of them developed leukemia. Scientists did tests and they discovered the problem. The therapy had worked. It had inserted a gene into the children's DNA, but it landed in the wrong spot. Right next to it was a cancer gene called LMO2, which was accidentally switched on by the treatment, ultimately driving the development of leukemia. These tragic setbacks brought the entire field of gene therapy to a halt for nearly a decade. But it also taught scientists 2 important lessons. Number 1, we cannot use full viruses to deliver genes to a patient, as we can have severe side effects. And number 2, we need to be careful where the gene is placed in the patient's DNA. Any mistake can have fatal consequences. So scientists went back to the drawing board. They began developing safer delivery systems, such as the adeno-associated virus. Instead of using entire viruses, These systems rely on only parts of the viral machinery to deliver therapeutic genes into cells. As a result, immune responses in patients were significantly reduced. Simultaneously, researchers started searching for ways to place genes more precisely within the patient's DNA. And this led to the development of the first generation of molecular scissors, including zinc finger nucleases and TAENs. These tools enabled for the first time to deliberately cut DNA at specific locations and deliver the right genes. These were major breakthroughs, but the technologies remained difficult to use. Designing these molecular tools was expensive, slow, and took often years for only one proof of concept experiment. It seemed like gene therapy is some technology from the future, until one discovery in one of the tiniest organisms on Earth changed everything. 1987. Osaka, Japan. A research team studied DNA sequences in bacteria. What they found was peculiar. A series of short repeats in DNA, the same sequence over and over again, separated by unique sequences. No one knew what it means, but having the same sequences over and over again must mean that there is an important function associated to it. A few years later, scientists discovered that these sequences actually match the DNA of viruses known to infect bacteria. Somehow, pieces of viral DNA had become part of the bacterial's DNA. It became clear that these repeating sequences in the bacteria's DNA must have some important role, but it would take years more to finally uncover it, leading to one of the greatest discoveries of humankind. 2007, France. Scientists at Denisco, France investigated why some yogurt-producing bacteria are resistant against viruses, while others are not. What they found is that bacteria can become resistant after a virus attack by integrating the virus's DNA into their own genetic information. And they placed the viral DNA right between the series of short repeats others have described. When a virus attacks a bacterial cell, it injects its genetic information into the bacterium. The bacterial cell has now some unknown mechanism that can take the DNA of the virus and integrate it into its own genetic information. If the virus returns, the bacteria now recognizes the infection and destroys the invader's DNA. The genetic region where viral DNA was stored was called clustered regularly interspaced short palindromic repeats, or CRISPR. A name that would go down in the history books. It just needed one last discovery. March 9th, 2011. San Juan, Puerto Rico. Scientist Jennifer Doudna and Emmanuel Charpentier walked through the old town of the city. Charpentier is a French microbiologist, and she was waiting for this moment. She had been hoping to meet Jennifer Doudna for days, because she had a request for her. You see, Jennifer Doudna is a renowned biochemist known for work on RNA molecules. Charpentier had recently finished a study on tracer RNA, a mysterious RNA molecule in bacteria that she suspected to somehow be involved in bacterial immunity and CRISPR. She also suspected that tracer RNA works together with a protein called Cas9. Cas9 is a protein that is known to cut DNA, but its relationship to tracer RNA wasn't known. Understanding how these mysterious molecules work together needed a brilliant biochemist. They decided to join forces to solve the mystery of tracer RNA and Cas9. June 8, 2012. After a year and a half of research, Jennifer Doudna and Emmanuel Charpentier were ready to reveal the breakthrough. Through a series of elaborate experiments, they finally solved the mystery of tracer RNA and Cas9. Cas9 is a protein that can make cuts in the DNA of an organism. For this, Cas9 uses two RNA molecules called cRNA and tracer RNA. Both of these RNA molecules guide the Cas9 protein to a specific sequence in the DNA. Once it arrives, Cas9 cuts the DNA. In bacteria, tracer RNA carries copies of viral DNA sequences. So when a virus attacks, tracer RNA guides Cas9 to the viral DNA, and Cas9 cuts and destroys it. And this is the mechanism behind CRISPR and bacterial immunity. That was already a big discovery, but Doudna and Charpentier didn't stop there. They realized that tracer RNA and cRNA could be fused into one molecule called guide RNA. And this small change made the whole system so much simpler. We now have a system that only has two components, the Cas9 protein and guide RNA. With these two components, we can cut and edit the DNA of any organism we like, and this includes humans. In the following year, multiple research groups showed that CRISPR-Cas9 could actually be used for gene editing. Researcher Feng Zhang in this group demonstrated that CRISPR-Cas9 can change human DNA. His team successfully cut the DNA of human cells and even inserted new DNA sequences into their genetic information. Simultaneously, the laboratory of George Church showed that CRISPR-Cas9 could cut DNA with remarkable accuracy in human cells. Shortly after, a South Korean team and Jennifer Doudna published separate studies confirming that CRISPR-Cas9 can break human chromosomal DNA. And the implications were massive. Before, gene editing was expensive and took years in order to do one proof of concept experiment, and now that only takes months or weeks. CRISPR-Cas9 technology is fast, cheap, and extraordinarily precise. Within only a couple of years, CRISPR gene editing technology became one of the most adapted technologies in laboratories all across the world. It transformed biology almost overnight and earned Doudna and Charpentier the Nobel Prize in Chemistry in 2020. Although CRISPR-Cas9 technology is still relatively young, we've already seen first successes in clinical trials. In 2018, CRISPR-Cas9 was used to treat patients with beta thalassemia. Beta thalassemia is a genetic disorder that is caused by changes in the HBB gene. When the HBB gene mutates, the body cannot produce enough functional beta globin, and patients have less functional red blood cells. To treat beta thalassemia, the doctor removed cells from a patient, edited them using CRISPR-Cas9 in the lab, and then reinfused the cells. And this approach was very smart. By taking cells out of the body and growing them in a petri dish, scientists can make sure that the therapy reaches the right cells. And they also can verify that the correct genetic changes have been made. And this makes the whole procedure very safe. The results were remarkable. Patients who had needed lifelong blood transfusions became transfusion independent. In 2024, this therapy, marketed as Casgevy, became the first CRISPR-based treatment approved by the US Food and Drug Administration. Of course, many genetic diseases cannot be cured by simply taking out cells and then editing them in the petri dish and then putting them back in. That mostly works for some blood diseases. And this is why scientists are not directly working with delivering genes with gene therapy right into the body. In 2020, a patient was treated with a CRISPR therapy for an inherited form of blindness. The treatment involved injecting CRISPR components into the eye to remove a disease-causing mutation. Astonishingly, some patients experienced measurable improvements in vision, proving that gene editing in the body is possible. But CRISPR-Cas9 gene therapy is not without risks and challenges. We need to deliver the DNA to the right cells, and we need to make sure that no other unintended changes are made to the cells. If we make cuts in the DNA in the wrong places, then we can have the development of other diseases, such as we've seen in past trials. And this is why right now many scientists are working towards making the treatments even safer and more effective. With the immense possibilities of CRISPR-Cas9 comes profound ethical responsibility. CRISPR-Cas9 treatments are currently restricted to cells outside the reproductive system. The reason is that any changes made to reproductive cells would be inherited by future generations, which could be dangerous if unintended genetic changes were introduced. Unfortunately, these ethical boundaries have already been crossed. In 2018, a Chinese researcher announced the birth of the world's first CRISPR-edited babies. He claimed to have altered the DNA of twin girls to make them resistant to HIV. The experiment was highly controversial and condemned by many prominent figures in the field, including Jennifer Doudna. This story is actually a very deep rabbit hole, as leaked documents suggest it may not have even worked. We've covered the full timeline and the leaked documents in a past video if you're interested. And there is also growing debate whether this technology could be misused to not treat genetic disorders, but instead change human traits such as eye color, height, or hair color. Such possibilities raise serious ethical questions regarding designer babies and how far we should go into rewriting our own DNA. For billions of years, our DNA shaped our health and biology beyond our control. Now for the first time, humanity holds a tool capable of rewriting the genetic instructions of life. What began as a curious observation in bacteria has evolved into a powerful platform for treating genetic diseases. The gene editing revolution is no longer theoretical. It has already begun, and its consequences for medicine, society, and our understanding of what it means to be human are just starting to unfold. And this could be one of the most consequential technologies humanity has ever developed. I hope that you enjoyed this video, and what's your opinion? How can gene therapy benefit humanity, and do you have any ethical concerns? Let me know in the comment section, I'm interested to hear your opinion. And with that, I'll see ya. If you're interested in a CRISPR gene editing scandal or a medical case where a man lived with almost no brain, you might like these videos.